So, good morning. I'm very happy to give the introduction of this summary of the second Gloabl Summit on Precision Psychiatry, which is organized in Brussels, where we are since May 15. It's the last day of the meeting. My name is Marion Le Boyer. I'm a psychiatrist by training. I'm professor of psychiatry at the Paris-Est University in Créteil, where I'm co-chair of the Department of Psychiatry of Mondor Hospital. I'm also head of the INSERM laboratory, which is called Translational Neuropsychiatry Laboratory, and I'm CEO of two French organizations which are supporting this summit. The non-profit foundation called Fondation FondaMental, and the French program on precision psychiatry that was selected by the France 2030 program. Maybe I should take this to move the slides. So the goal, the aim of the summit was to discuss goals, successes, and challenges of precision psychiatry with all stakeholders from regulatory and funding agencies, patient and caregiver associations, policymakers and decision makers, industry, academia, and international associations, with you journalists, of course. We have here in Brussels more than 3 international experts who are present with us to present the field of precision psychiatry. And we had, during the past 2 days, more than 400 participants online from the overall globe. In fact, we had lots of people coming from everywhere at the international level. This summit follows several meetings with all have in common to share data and vision regarding this field of precision psychiatry. So the first meeting was held a year ago in Switzerland, in Caen, where we had a think tank on precision psychiatry organized by Fondation FondaMental, with at the end a white paper written and several consensus papers. Then we had a second meeting in Nice, in France, organized by ECNP, where we discussed again this precision psychiatry field, and where we wrote and are in the process of writing a roadmap for precision psychiatry. And then we have this meeting, which is ongoing, with the as outcome, on top of this press conference, we're going to write a policy brief for Europe institutions, and we're going to write several papers out of this meeting. So why do we need precision psychiatry? We just want to remind you that mental disorders are one of the greatest challenges of the 21st century, as shown on this slide. It is really a major public health issue, as shown by the fact that major psychiatric disorders are among the top 10 leading causes of burden worldwide. It affects an estimated 800 million people worldwide, which means that 10% or above of the planet populations are affected. It's also associated to an enormous economic burden, almost 4% of a nation's GDP, and in particular in France, the teams of Fondation FondaMental have recently published that the direct and indirect cost of mental disorders is up to 160 billion euro a year. It's also the first cause of DALYs being the number of days lost because of a disorder, and WHO has demonstrated that it is the first cause of acquired handicap. However, despite this burden, there is still a high unmet need because diagnoses are still either delayed or underdone. These disorders are at best partially treated and often with side effects, and there have been very few innovations in the field in the past 50 years. So we really need to do better. We do believe that the situations we are facing are explained by failures of clinical trials, which are explained by 3 reasons. First, the diagnostic classification we are using yields very good inter-rater reliability between raters, but no biological validity. And they also yield the identification of very heterogeneous clinical entities and overlapping clinical entities. The second problem we're facing with clinical trials, which explain failures again, is that the endpoints we're using are not objectively measurable and very often not specific to any causal mechanisms. And last problem, the treatments being tested in the past years for in clinical trials are usually not mechanisms-based, and most of the drugs which have been discovered in the field have been discovered by serendipity. So the good news is that precision psychiatry brings to the field some rules which are exactly what we need. First, they require that treatments have to target a specific etiological mechanism. Second, they require that patients included in the trials have to be defined and belong to specific subgroups, and patients have to be diagnosed with biomarkers. And three, the measure of efficacy, of course, need to be objectively measured and targeting what the treatment targets precisely. So the goals of precision psychiatry is to be able to identify the right treatment for the right patient at the right time. This is a cartoon to represent the vision we have for precision psychiatry, where we want to improve measurements and stratification of the categories we have today, and we thus want to move away from what we're doing today, which could be described as one size fits all, to something very different, which we could call haute couture à la française, where we could try to identify for a particular patient or a particular subgroup of patients exactly the size or the color that they need. The good news is that now we have tools to pursue precision psychiatry. We can move on from what we have today, which are this international classification, which describe heterogeneous diagnostic entities, overlapping entities, which are mostly defined by behavioral symptoms, and we can use what we call deep phenotyping to, based on the thorough assessment of patients, of course with clinical data, but enriched with exposome, digital phenotyping, omics, brain imaging, and this should help us to identify homogeneous clusters of patients who share common transdiagnostic signatures. And this should help us to move towards the identification of mechanisms, circuits, and systems to target the underlying biology linked to maybe specific symptoms or specific dimensions, aiming at precision psychiatry, again, being able to identify subgroups and having mechanism-based treatment. So we have rules for precision psychiatry that have already been alluded to, but we also have challenges in front of us, and the challenges are described here on the right. First, we need to demonstrate the validity of the stratification biomarker, but then we also need to obtain regulatory approval of these biomarkers. We also need to demonstrate the validity of the endpoints we want to use in the clinical trials, but here again, we need to have approval of regulatory teams or tools. And three, we need to demonstrate the clinical efficacy of mechanism-based treatment, and of course, to get regulatory approval. So I just want to finish with two slides in my introduction to present the two French organizations which were the hosts and organizers of this summit. First, the Fondation FondaMental, which is a French non-profit foundation, which was created by the French government in 2007 to support research and innovation in the field of mental health. We're dealing with major psychiatric disorders. We have identified more than 20,000 patients in our network of expert centers, and a little bit above 8,000 are now in dedicated and shared databases and biobanks. We work in networks with academic labs. We have developed digital platforms and apps. We are coordinating a network of 53 expert centers for major psychiatric disorders, all located in different places in France. We have been able to create very large cohorts of patients with psychiatric disorders. We are coordinating more than 40 research projects, and we are working with several small and big pharma and startups in the field. And just to conclude, just want to briefly present this program project in Precision Psychiatry that was funded by France 2030, which is based on the creation of the very large cohort, which is going to be called French Minds, of above 10,000 patients, including patients with bipolar disorders, schizophrenia, major depression, and autism, identified through the centers, expert centers, and centers from RHU Psycare all through France. And all these patients will follow the rules of deep phenotyping. They will be thoroughly assessed with clinical phenotypes, with genomics, metabolomics, proteomics, epigenetics, and immunology. They will be assessed regularly with digital tools and with brain imaging, MRI, spectroscopy, and electrophysiology. And the goal in parallel is to be able to explore in animal models the mechanisms underlying the biomarkers we want to search for, to perform clinical trials with specific mechanism-based treatment for specific subgroups identified by the biomarkers we're looking for. We also want to stimulate private-public partnership, and of course we want to contribute to destigmatization and information by engaging users, healthcare professionals, and policymakers. Thank you very much. And I will pass the word to Professor Andy Miller, who is going to introduce himself, maybe better than I can do.
Sure, maybe.
Thank you.
My name is Andrew Miller. I'm a physician scientist, psychiatrist at Emory University in Atlanta, Georgia. I'm a professor of psychiatry and behavioral sciences at Emory and the vice chair of research at Emory. And this morning, I'd like to talk about how we can use precision psychiatry to target the immune system and inflammation to treat depression. So you have the slides there. So I think it's important that we have a little context here. And in psychiatry, we've not done a really great job in terms of coming up with new medications in terms of treating various psychiatric disorders. In fact, psychiatric medications have not meaningfully changed in over 50 years. And I'm being generous there. It's probably closer to 75 with some minor modifications having to do with tolerability and safety. And as Dr. LeBoyer mentioned, most of these drugs were developed or discovered, and most of the drugs that we use today in psychiatry were discovered by serendipity, by chance, and they were not discovered based on any understanding of the mechanisms of psychiatric disease. And what that has ended up with, excuse me, is a series of medications that are quite nonspecific in their nature. We have a number of them, and we use them for everything. So if you look, you can see antipsychotics. We use those for a number of psychiatric disorders. Same thing with antidepressants and mood stabilizers, and use them pretty much indiscriminately across various psychiatric disorders. And while we know a lot about the pharmacology of these drugs in vitro and in laboratory animals, we have very little information of how these drugs work in humans, what the mechanism of action is in people. And I think one of the results of this sort of non-specificity in terms of our currently available treatments is we have a number of individuals who are not able to take advantage of our conventional treatments. And in fact, a third or more of depressed individuals, for example, are non-responsive to our conventional therapies. And that includes not only the pills that I've indicated here, but also psychotherapies and other treatments. And that's sort of where precision psychiatry comes in, because clearly what we understand now is one size doesn't fit all. And when we look at various psychiatric disorders, we realize that they are made up of very heterogeneous populations of individuals, as you can see here. And in precision psychiatry, what we try to do is take those heterogeneous psychiatric disorders and sort of break them down using a variety of different strategies that Dr. LeBoyer mentioned, where we're looking at various biomarkers, we're looking at various neuroimaging circuits in the brain, we're looking at genetics, we're looking at different symptom profiles, and based on these various assessments, we can break the patients down into a variety of different clinical subtypes or biotypes here. And from the quote here, you can see on the right-hand side of the slide, at its core, when we're talking about precision psychiatry, we're talking about focusing on understanding the neurobiological mechanisms of all these different biotypes that when we break down these disorders, and understanding those mechanisms allows us to begin to develop personalized treatment options, as indicated at the bottom of the slide there. So one of the mechanisms that we've been quite interested in is a pathway to pathology in depression, which is inflammation. And I think many of you know that inflammation is our body's natural response to infection or wounding whenever there's tissue damage or destruction. But in modern humans, we have a little bit of a problem with chronic and persisting inflammation. And when this occurs, it can do damage to many parts of the body, affect many parts of the body, including the brain. And this has been known for quite some time. The front of Time magazine, The Secret Killer, inflammation playing a role in heart disease, cancer, and also diseases involving the brain, including neurodegenerative disorders, and of course, our interest, psychiatric disorders. And this role of inflammation as a common mechanism for many diseases in modern humans was considered one of the insights of the decade by the journal Science in 2010. So we've recognized this for quite some time. And you might ask the question, well, where is this inflammation coming from? Well, we know quite a bit about this, and probably the biggest offender is obesity. So fat cells grow very quickly, they outstrip their oxygen supply, and they die. And the body sees this as a wound. And so fat tissue is loaded with inflammatory cells, immune cells from the immune system that are producing high amounts of cytokines that can get into the brain and also change metabolism throughout the body. And many of those metabolic changes also occur as a function of aging. And there's a process called inflammaging. As people get older, they're more vulnerable to an exaggerated inflammatory response. Also, stress, especially early life stress, chronic stress, can change set points in the body that are important in regulating the inflammatory system, the immune system. You've heard a lot about the gut microbiome. There's dysbiosis, and microbial products from the gut can enter into the bloodstream and activate the immune system and lead to inflammation. I mentioned many of the chronic illnesses that people suffer from are also associated with inflammation, as are chronic infections. And then, of course, we do a number of things to people. And this, I used to work in the cancer center, so particularly sensitive to the surgery, radiation, and chemotherapy, as well as some of the immunotherapies can lead to chronic inflammation. And as you well know, there are many changes in behavior that are a consequence of having and being treated for cancer. And in terms of understanding how inflammation affects the brain, we know quite a bit about this. We know the changes that occur in the immune cells themselves, the immunometabolic changes that occur, and they occur both in the cells themselves as well as in the body in general. We know that the immune system, once activated, produces a variety of inflammatory molecules that we know can get into the brain and change neurotransmitter metabolism, affecting dopamine and glutamate, and then ultimately affecting neurocircuits in the brain that are playing an important role in terms of motivation and reward processing, psychomotor speed and cognitive function, as well as the sensitivity to threat. And these changes that occur in circuits also lead to changes in behavior. And what's interesting about understanding this mechanism from the immune system all the way to behavior is that we can then develop specific targets and in particular treatments that target these various aspects of the mechanism by which inflammation affects the brain. So starting with immunometabolism, there are a number of drugs that can be used to target changes in metabolism in immune cells, as well as a number of drugs that can affect inflammation itself. We can go after the neurotransmitter changes that occur as a function of inflammation, drugs that target dopamine as well as glutamate. We can manipulate the circuits that are altered as a function of inflammation through therapies that involve neuromodulation. And finally, based on the changes that occur in the circuits and ultimately the behavior, we can use various psychotherapies that target the effects of inflammation on the brain. And these are all specific to this mechanism by which inflammation is affecting the brain. Now, I just should make the point, and this is all what precision psychiatry is all about. Not all depressed patients have increased inflammation. Depression is not an inflammatory disorder. About 30% of patients have increased inflammation. So we need biomarkers to identify, well, who's inflamed and who's not. And surprisingly enough, that's not as difficult as it may seem, in that there are available biomarkers that are in the clinics today, and at least in the United States and in most all clinics and hospitals, you can have C-reactive protein measured in your blood. That's produced by the liver in response to inflammatory mediators. And according to the guidelines from the American Heart Association, Centers for Disease Prevention and Control, we know what is low, medium, and high level of inflammation. And that allows us to know whether or not a patient is inflamed and can inform us whether this might be a particular patient who might take advantage of or benefit from some of the targeting and treatments that I just mentioned. And in order to test those various treatments, we need to have a strategy, and the strategy is born out of precision psychiatry. So this is precision psychiatry in the making. And based on all this knowledge that we've been talking about at this meeting and that we've learned about a precision approach, is that, and Dr. LeBoyer mentioned many of this, our trial designs have to be different. They have to change. We need to enrich our sample for patients with increased inflammation, if that's the mechanism that we're targeting, because inflammation is not for everyone. We have to design our outcome variables that are tied to the effects of the inflammation on the brain. So we have to do the outcome variables tied to the mechanisms that we're targeting. In this case, anhedonia, anxiety would be important targets. And finally, we have to determine, if we know the mechanism or think we're targeting a mechanism, we need to determine that the drugs that we're using are actually hitting the target and show that the decreases in inflammation, for example, in the brain or the blood are correlating with the changes that we see in behavior. So we can trust, but we need to verify that, in fact, we're hitting the target. So I think the time is right. Based on understanding mechanisms, we can use intelligent trial design. We can enrich for the patient populations that have the mechanism of interest, the biotypes that I was alluding to before, and we can align the outcomes and the targets with, in this case, the effects of inflammation on the brain, and in this way lead the charge in precision medicine and psychiatry. So I'll stop there. Pass it on to Martine.
Thank you, Andy. Good morning, everybody. My name is Martin Kas. I'm a professor of behavioral neuroscience at the University of Groningen in the Netherlands. I'm also the ECNP president, which is the largest organization in Europe for neuroscience applied research. And I will be presenting today on the work we have been doing in the PRISM project, which is an EU-funded project. My next slide will come. Oops. I will be presenting today as the project coordinator of the PRISM project that started about 8 years ago, where in contrast to the work of Andy looking at depression as a single diagnostic criteria, we actually try to look at the biology across different disorders. So really trying to address the transdiagnostic aspect, and specifically zooming in on social withdrawal. So in our daily lives, the level of social interaction is visible at many different developmental stages, from the very early stage where there is mother-child interaction, to the play behavior, to our group behavior, and subsequently the partner choice. And as Erik Erikson already indicated, this is a quite critical sequence of events for actually psychosocial development as well, where there is buildup of trust, autonomy, identity, and intimacy. So clearly these highly integrated social behaviors are critical not only for biological reproduction, but also actually for our psychosocial development. Interestingly, we do see very similar patterns of behavior, not only in humans, but also in the animal kingdom. We can actually see those very similar behaviors at the very different behavioral stages, ranging from insects all the way to birds and other mammals. So this is clearly a very conserved phenotype. Sorry about the delay in the slides here. And what is important is that we do not only see that at the behavioral level, but there is actually a whole scheme of biological functions that are developed across development and that are critical for these different stages. So, for example, we see early in life the development of our sensory systems, which are, of course, critical for the mother-child interactions to pick up on these social cues. Now, if we then go to social dysfunction, like social withdrawal or social avoidance, we can actually see that that is present in many different psychiatric and neurological disorders. So actually social functioning or social dysfunctioning is a transdiagnostic phenotype that actually goes beyond the current diagnostic criteria. So one could wonder where actually this social dysfunction has a common biology. If you think about very early developmental disorders, such as autism spectrum disorder or schizophrenia, which usually starts around puberty, or even in Alzheimer's disease, which we see later in life. So is there maybe common biology across these very different disorders? So what PRISM tried to do here is actually go beyond the current diagnostic silos and ask the question whether maybe social dysfunction, irrespective of the diagnostic criteria, has some biological basis. And this is the clinical trial that PRISM has set out and tried to show proof of concept, where we studied patients with Alzheimer's disease and schizophrenia in the same trial, assessing biological data going from genetics to neuroimaging and behavior, and see whether we actually could produce homogeneous populations based on this biological data and irrespective of their initial diagnosis. And this was a large effort with many different parties involved, including big pharma, medical centers, small, medium enterprises, patient organizations, and also ECNP as our disseminator of our activities. Now, PRISM not only focused on social withdrawal to give you the full picture, but also on domains such as sensory processing, working memory and attention at various different levels, from biology all the way up to behavior. And we did this both in humans and in rodents, because we think it's important that we can use the rodent studies to back translate the human findings and test for causality. We also introduced novel digital technologies because we think it's important to assess social functioning longitudinally, objectively, and quantitatively. So alongside the well-known questionnaires for social functioning, we also use this digital tool, BHAP, to come forward with novel digital endpoints for social functioning. And then just as an example, here you see that we were able to show transdiagnostic digital signatures across these patient populations that actually show very different social patterns, irrespective of the diagnosis. Now, the question, of course, is there then a transdiagnostic relationship between social functioning and integrity of a specific brain network? And actually at the level of wide matter tracks, the genetics, as well as fMRI connectivity measures, we figured out that the default mode network, which is a basic network in the brain, and the connectivity within that network is actually transdiagnostically associated with the levels of social functioning, irrespective of the diagnosis. So we found this across the population. So when social functioning is low, then actually the connectivity in this network tends to be low as well. And this is actually the first proof of concept that you can actually address this transdiagnostic approach across very different disorders, showing social dysfunction actually at the biological level. So we also have, as I said before, an animal preclinical testing battery, and we did some data mining on social functioning. For example, we showed in a genetic study that there were specific independent loci associated with social functioning. And we have integrated now animal studies where we can actually follow animals and actually measure their social functioning 24/7 to actually validate these findings. So what PRISM has shown, I think, is that we showed a proof of concept of a transdiagnostic clinical study, and we identified quantitative biological parameters related to social functioning and neural integrity, irrespective of traditional diagnosis. We delivered a testable hypothesis for the understanding of the neurobiology of social dysfunction. I think we offered clinical practice and drug discovery research insights. And we have developed an informed preclinical testing battery homologous to the assessments in the clinical study. And the digital tool we have developed is now initiated for a EMA qualification procedure to realize a qualified digital endpoint for social functioning. Now, the future outlook, I think we're really setting the stage here to establish a biology-based framework for mental disorders to bring the right treatment to the right patients, but I think that we should enhance our fundamental biological understanding of mental disorders etiology and provide transdiagnostic biomarkers and predictive model systems to accelerate the treatment development for patient-relevant functions beyond the current diagnostic criteria. Thank you.
Yes, hello, I'm the last speaker. I'm Brenda Penix. I'm professor of psychiatric epidemiology at Amsterdam UMC in Amsterdam in the Netherlands. And I'm actually going to add, I think, an example by really adding data and give you a glance of what it means to be talking about precision psychiatry. I'm doing that from the example of immunometabolic depression. And that is because depression is actually one of the most common conditions, and that is the condition that I have been examining in the last 20 years. So if we talk about depression, then we talk about a really heterogeneous condition with lots of emotional, cognitive, but also somatic symptoms that are accumulating in a person and are really having a chronic impact on the life of a person. And unfortunately, I think we are facing, as a society, unfortunately, an increase in major depressive disorders in the current time. Depression is, as you compare it to many other somatic conditions, one of the prime drivers of disease burden, as also the WHO calculates every few years. I think currently depression is listed as the second condition, and it's expected to be the first condition with the largest impact in disease burden in 2030. As already mentioned before by, I think, all three speakers, is that we are really aware of the fact that heterogeneity in etiology and symptomatology is having a major impact in the way we look at conditions, but also how we should treat them. And I think this is especially clear in the field of depression, as mentioned already by all people here. One size doesn't fit all. We know that antidepressants are effective, but unfortunately they don't work in all people. And we really lack treatment that are more directed at biological mechanisms. So this is, again, another picture of that we have described of how precision psychiatry looks like. We really need to move away from heterogeneous depression groups through measuring the biological basis behind disorders, but not just only the biology basis, but also incorporate life experience and lifestyles, for instance, in better quantifying and phenotyping our patients so that we can really use that information to arrive at more homogeneous depression profiles. So I also want to come back a little bit to the relevance of the immune system in depression. And very often people ask me, is that really a driving factor, or is it just a consequence of depression? I think there's really ample evidence that the immune system involvement in depression is really contributing to the experience of depressive symptomatology. And I think that comes really from different types of evidences that we have in the literature. We know that people with autoimmune diseases and infections, for instance, have an increased risk of developing depression. We know that if you experimentally manipulate the immune system by, for instance, vaccines or endotoxin administrations, which has been done, yields symptoms that are really very much in line with depressive symptomatology. We know from many studies that if we compare blood levels of immune markers between people with depression and without depression, that on average people with depression have higher levels. And also, I think there are lots of developments in the genetics fields that really tell us more about the genetic basis of depression. And if you look at the huge studies out there where they have looked at the genetic basis in more than millions of people, it's very crystal clear that inflammatory genetic pathways are among the top hits of genes that are relevant for depression. I think, Dr. Miller really clearly indicated that already, that inflammation is a bodily process and it's connecting to all kinds of activities, including lipid metabolism, for instance, at very many peripheral tissues. So inflammation is really a systemic condition of the body, including the brain. And that's why I also call really depression, and not just a brain condition, as we already talked about, but really as a condition that is impacted in the entire body. And it's not surprisingly that we therefore also see that there is a high comorbidity between depression and somatic conditions, which actually is true for many other mental conditions. Unfortunately, we know that mental conditions are increasing the risk of developing somatic health conditions and unfortunately also lead to an increased risk of dying earlier due to somatic conditions. And I think immunometabolic dysregulations are really playing a big role in that. So one of the things we are doing in the Netherlands is we're studying large cohorts of depressed patients over time. And that I think is really useful because by getting lots of people with depression and also controls following up over time, we can really study the course patterns and we can really study the development of somatic conditions, but we can also define and look at etiological processes, which we do in the Netherlands study of depression and anxiety. And one of the findings that comes from the depression study is that we can really zoom in on more than 900 people with depression and really identify who actually are the ones who are then showing immunometabolic dysregulation. And as Dr. Miller already clearly indicated, we should really be making certain that we're not assuming that everybody has the same dysregulations, because we know that is not true. So what we do see in our cohort is that there is about 1/4 of all persons with depression that has this profile that we start to call immunometabolic depression. And these people, they are the ones who have most evidence for immune dysregulation, inflammation, dyslipidemia, but also conditions like leptin and insulin resistance, which, for instance, are really underlying disease conditions like metabolic syndrome, clustering of cardiovascular risk factors, and the development of cardiometabolic conditions. So it's not everybody, it is really a subgroup. And that subgroup can also be identified by specific symptoms that are different in this subgroup. So by nailing down and really looking in detail at how do these people look like, we do see differences in the type of symptoms that people with immunometabolic depression have. They have more energy-related atypical symptoms of depression, such as an increase in appetite and weight during their depressive episode. They have more fatigue, and they, for instance, have hypersomnia. So you can wonder, why is this relevant? Why do we need to study that? And I think Andy Miller already clearly indicated that there is some evidence that, especially when we target that specific group with depression, there is indications that these people are responding better to anti-inflammatory medication. So now we have a condition that we can specifically target with anti-inflammatory medication. And this is obviously something that we need to test further at a larger scale, which we are currently doing in various studies in Europe and in America. But I think it's also giving us examples where we can really target and examine the impact of lifestyle, because we know that lifestyle is very important for immunometabolic dysregulation, and there are ample studies that show that exercise interventions, for instance, are beneficial for depression and are really beneficial also for decreasing immunometabolic burden. That's one study that we have done in Amsterdam quite a few years ago now, where we have compared the effect of running therapy with the effect of standard antidepressant treatment in people with depression. And as you can see from this slide, exercise therapy versus SSRI treatment were as effective in reducing depressive and anxiety symptoms over time. So they are both tools and interventions that we can use to reduce the burden of depression and anxiety symptoms. However, they may do that through different processes. And if you look at other outcomes, such as immunometabolic health outcomes in these same individuals, we do see that actually it were the running therapy persons that either improved their immunometabolic health function, and the people on antidepressants who actually did, in certain aspects, worsen in terms of their immunometabolic health profile. So I think this also shows that by comparing our interventions and looking at impact, not just only on symptomatology, but at really underlying mechanisms, we may learn better what are the working mechanisms and who should be getting what kind of interventions. So my last slide, I think this is an example of how, by further zooming in on underlying pathophysiology in subgroups of specific patients, we can arrive at better knowledge and better insights, but also more targeted interventions. And this is just an example. I think our field is ready for getting more of these examples. And I think there is now more and more awareness and consensus that we really need to take this route of precision psychiatry by focusing on biologically informed treatments and classifications. Thank you very much.
Thank you very much. I think we are ready for questions, if you have any.
And depressive patients have information. What is the ratio, the observed ratio in general population?
And did you want to answer?
Depends on the population, obviously, and so if you're, it's like the question, what is normal? And the data show that somewhere around 15% If you looked at large populations and you pooled everyone, but everybody, has issues. I mean, if you look at medically ill patients, obviously the inflammation goes up, as does the rates of depression. But in the general population, we're talking about somewhere in the neighborhood of 15% would have increased inflammation.
Right. So there's Lee Du Guan from agency Press Media AP News. Professor Cass said the PRISM result offered insight in new drug discovery, but I said about social dysfunction, what about other type of treatment? First question. Second question, cognitive behavioral therapy or other psychological methods, neuromodulation techniques. Do your results would also help understand how those other kinds of treatments?
Thank you for that question. That's of course the ultimate aim. So but we realize we first need to get a better understanding of those transdiagnostic relationships between biology and brain function. Once we know the circuits that are underlying these different patient groups, we may be able to find targeted treatments, either as indicated in the examples. It could be a behavioral activation therapy, for example, in very low social function individuals, or maybe we're able to find compounds that can target specifically these neural circuits at moments of low social functioning. Or maybe the technology or indeed of brain stimulation is actually optimizing currently very rapidly. That could potentially be a way forward as well, but that's of course still early days. But the PRISM project really tried to provide proof of concept of this transdiagnostic relationship between brain function and biology. And based on that knowledge, we can of course start to identify particular interventions to help those who are in need.
Thank you. So there's another question. Physical activity could target better the inflammatory subgroup of the depressed patients? Question mark.
Yeah, I think that question mark is adequate, because I think that's also the question mark I still have. I don't think we have large enough exercise studies out there in a psychiatric population that we can look at to really see whether we can stratify patient groups and see whether one group responds better on exercise intervention than the other. I think that is too early, because we really have not enough large-scale interventions that we can use to test this question mark. But I think it is a very logical expectation that if you expect that exercise interventions are reducing immunometabolic stress, then that is especially beneficial for people that start out with high immunometabolic stress.
Thank you very much. This is a question from a clinical perspective. So what kind of diagnostic testing and treatment option could we look forward to now to detect and address inflammation while managing depressing disorder in our patients?
Okay, I'll take that one. Actually, we're progressing fairly rapidly in that space. We have clinical trials underway identifying patients with increased inflammation and decreased motivation. And we're treating these patients with drugs that replace dopamine, because inflammation targets dopamine in the brain. And interestingly enough, when you give drugs that promote dopamine, in this case, we're using a drug that's used in Parkinson's disease, which is a disease of loss of dopamine. When we replace dopamine with a drug called levodopa in depressed individuals, we see that there are circuitry in the brain that involves disruptions in circuits that regulate motivation and motor activity and cognition. That replacement of dopamine can reverse those changes in the circuitry and reverse the changes in behavior, particularly motivation. So that's one way we're approaching it. And so that may be ready for prime time sooner rather than later. And also we're looking at anti-inflammatory drugs for the treatment of this subgroup of patients. And there are a number of efforts that are going on across the world where people are looking at different anti-inflammatory drugs, and we're certainly part of that as well. And you know, we're not looking too far away in terms of those kinds of treatments and having the kind of data to support whether or not we should be thinking changing guidelines or coming up with indications for drugs that might target this inflammatory subpopulation.
Thank you very much. There's another question. That's what about inflammatory biomarkers? And I quote, CRP is known in rheumatology, infectology, not to be the better marker. So what about using a panel?
Yes, I think that's a very appropriate comment. And I think that's really much how we think about that. And that's also why in lots of our research studies, we are actually now going beyond CRP and looking at larger panels of inflammatory sets. Because yeah, I think we really also need to dive further into, is there another set of biomarkers? I think it's very unlikely that maybe one biomarker itself may be enough to completely best predict who is best responding on anti-inflammatory medication, for instance. But I think this is really something that we are doing right now and that we are really scaling up also in lots of studies also together. So there are now people really with data on anti-inflammatory medication working together, looking at the banked samples, for instance, and going back to measure more biomarkers than just CRP.
Thank you very much. And just wait a few more minutes if there's more questions from journalists.
Okay, well, thank you. I just want to thank you for having attended this press conference. I hope we have raised interesting questions and we're ready for more answers if you have next discussions and questions for us. And I want to thank very much our speakers this morning, in particular Brenda Penix, Martin Cass, and Andy Miller, to have accepted to describe some of the elements that were described during this second Gloabl Summit for Precision Psychiatry here in Brussels. Have a good day, and thank you again.